Hi everyone. So we're going to be talking about your Unit 6 progress check from AP Classroom, specifically question 1 of the FRQ. I'm going to take you through the question, try to explain the question a little bit better to you. This was a very complicated question, so I am going to assume that a lot of you guys are going to have a little bit of problems answering this question. So as I'm explaining, please try to follow in the explanation, pause the video whenever something is not completely clear to you. Try to check it out as much as you can with the evidence from the question. And once you're done looking, or simultaneously, I don't mind, as you're looking at this video, you're also going to be completing a Schoology assignment where you are going to be scoring your own answers. So you should have finished this FRQ by 11:59 p.m. on Sunday. So today you need to be grading your own questions based on the score guide that I'm going to show you right now. First, I'm going to actually just talk about the question. I'm going to read the question out loud, and I'm going to talk about key things that you should be identifying in these sort of questions, and then I'm going to show you how to score each question. I'm also going to post this PDF on your assignment so that when it comes time to score yourself, you'll be able to check this PDF out and make sure you're scoring yourself correctly, and you're also going to be reflecting on the places you made mistakes. Okay, so it says, read each question carefully. Write your responses in the space provided for each part of each question. Answers must be written out in paragraph form. Outlines, bulleted lists, or diagrams alone are not acceptable and will not be scored. Also, I've had some trouble with some of you guys that you are not making a space between question A, question B, question C, and question D. You need to make it a space. It should be in paragraph form. Each question is a separate paragraph. Okay, so please make spaces between these questions. I'm thinking that they're gonna take points off of questions that are not well organized and do not have spaces in between. So please don't suffer from loss of points because of something like that, okay? So it says CD3 is a signaling protein that is typically found only in the plasma membrane of immune system T lymphocytes. CD3 is composed of several different polypeptides, including a gamma chain, CD3, I'm thinking that's lambda, this sign. I'm not 100% sure what that sign stands for, but it doesn't really matter. Scientists analyze the promoter of this chain, of this CD3 chain, for regulatory sequences that might have positive or negative effects on expression of genes. So it's already telling you that you have a promoter of a chain of genes, okay? You have a chain of genes, and you want to know if that chain of genes has positive or negative effects on expression of the gene. So you want to know if there are areas that are coding for positive or inducing transcription, and areas that are coding for negative or repressing transcription. Okay? So it says the scientists cloned fragments of this gene that included the first transcribed nucleotides plus up to 789 nucleotides of upstream regulatory sequences into plasmids in which the gene for the firefly enzyme luciferase immediately follows the fragments. Okay, so they put this regulatory gene sequence into a plasmid, and right after this regulatory gene sequence, there was a sequence that coded for the enzyme luciferase, which is an enzyme of a firefly. Okay? The plasmids were then introduced into the line of T lymphocytes, figure 1, and the cells were allowed to grow for a short while. Because the regulatory sequence of CD3 gene immediately preceded the luciferase gene in the plasmids, the activity, either positive or negative, of the regulatory sequence affected the amount of luciferase gene expression by the T lymphocytes. So basically what it's telling you is that this insertion of this gene right next to this other gene, the firefly enzyme luciferase gene, will affect how much luciferase, the enzyme luciferase, is expressed or how much it is not expressed in these T lymphocytes. So there's a lot of components here that you need to consider. First, you are attaching a new regulatory gene into a plasmid. After attaching that new regulatory gene into a plasmid, you're also placing it right next to a gene that codes for an enzyme of luciferase. I'm going to tell you why that's important in a second. And you're placing that plasmid with these two genes in T lymphocytes. Okay? Luciferase catalyzes a reaction that results in the release of light and is responsible for the bioluminescence, light flashes, of fireflies. So this enzyme that you are kind of trying to check out how it's regulated by this chain of genes, this enzyme is super important because it produces light. So that already gives you a key point here. Why are we checking this chain and whether it has positive or negative effects on expression of gene right next to an enzyme luciferase? We're checking it right next to an enzyme luciferase because we can see how this enzyme is expressed or not expressed if we see that the cells are producing light or they're not producing light. So now you have a phenotype that you can relate to the regulatory gene. You can relate to it by seeing if it is in fact promoting or repressing the enzyme. Okay, so it says, we'll continue. It says, by quantifying the bioluminescence or luciferase activity in the cells, in the T lymphocytes, remember, the scientists were able to determine the effect of each CD3 gene fragment cloned into plasmids, figure 2, on expression of the gene. So we have two figures. Figure 1 is a summary of experimental procedure, a series of plasmids containing fragments of the CD3 upstream regulatory sequence cloned immediately before the luciferase gene were constructed. Each type of plasmid was introduced into T lymphocytes. The amount of luciferase produced by the lymphocytes was dependent on the regulatory sequence present in each plasmid. So basically what happened, you have this regulatory sequence, the CD3 gene, which is a regulatory sequence, and it's right upstream of a region that contains the luciferase gene. Okay, so this is the location that was used in the plasmid for the CD3 regulatory gene sequence. They're inserted right over here, and then the luciferase gene, which produces this enzyme of bioluminescence, is right next to it. Okay, so we can check out what's happening right over here, what is being promoted, what is being repressed, by checking out if the luciferase gene is actually being transcribed and expressed, and if there is bioluminescence. Okay, so then you produce this plasmid. Okay, you produce the plasmid. Remember, the plasmid is that segment of DNA, or it's kind of like a circular DNA that you commonly find in bacteria, and you can insert that into other cells so that other cells can also express certain genes of interest. So you insert that plasmid into the T lymphocytes, and you're trying to check out luciferase production, you're trying to check out bioluminescence. Okay. Figure number 2 is the analysis of the luciferase activity of T lymphocytes containing plasmids with different amounts of CD3 regulatory sequence. Names of the plasmids are shown on the left, included regulatory sequences, and the resulting luciferase activity are shown on the right. Error bars represent plus or minus 2 standard error of mean. The transcription start site is indicated +1. Okay, so this is pretty much telling you that you've got the promoter sequence of CD3, and then it's telling you the different fragments. Remember that you're cutting this sequence in different fragments, and those cuts are being inserted into different plasmids, and those plasmids are being inserted into different T lymphocytes. Okay, so this is telling you that it contains the promoter of the CD3 and it contains a fragment. I don't know if this will represent 789 base pairs or what this number represents, but it doesn't really matter. What it represents is that there are different lengths of fragments right after the promoting sequence of CD3. And then you can compare these CD3 promoter plus different fragment lengths with relative luciferase activity. So you can see here, and again, it doesn't really actually tell me what the units are of luciferase activity. So I'm just going to tell you the numbers, but you can see here in the fragment that contains 789, I'm guessing base pairs. The activity of luciferase was in 4. Okay. Sorry. 419 in this one produced a little bit less of luciferase activity, like around number 9. This one over here, PCD315, produced a whole bunch of luciferase activity, around 10. Okay, so you can see how each one of these fragments in different plasmids are producing a different reaction in luciferase activity in the T lymphocytes. Okay, so it says, identify let's start going into the actual scoring guide. Okay, so all of these are your questions, and this is kind of what you saw in AP Classroom, how you were able to answer this. Okay, so it says question 1. Sorry, let's go into question 1. I'm so sorry. Identify both the cellular component and the location of the component that is responsible for producing the luciferase protein from mRNA transcribed in the plasmid containing T lymphocytes. This is a whole bunch of wording to simply ask you, where is the protein being produced and what is it using to produce this protein? So if you go into section A1, you can see that the response indicates that luciferase is produced by ribosomes. Of course it's produced by ribosomes because ribosomes produce proteins. And where does it produce proteins? It can produce in the cytoplasm or in the cytosol of the lymphocytes. Okay, so this was simply for you to know where is this production of the protein occurring. Okay, so this is question 1A because there's the identify here. Okay, you identified, you listed it out, perfect. Then question A2 would be explain what dictates to the lymphocytes that the correct order in which amino acids should be linked. Explain what dictates to the lymphocytes the correct order in which amino acids should be linked to form the luciferase protein. Again, that is fancy wording to simply ask you, how do you read an mRNA and produce amino acids from that mRNA? And you guys know this. The response indicates that the sequence of nucleotides in the mRNA, the sequence, dictates the amino acid sequence of luciferase. How? With triplets of nucleotides called codons. Each codon specifies for each amino acid to be added in order to the growing polypeptide chain. Okay? So that was, if you got that wrong, it's because you were overthinking it. It was asking you simply ribosomes and codons, not much more than that. Question B says, identify the independent variable in the experiment described. Identify the plasmid that was used as a negative control for luciferase activity. Justify including the plasmid with non-CD3 active promoter in the experiment. So this would be non-CD3, this one. This plasmid right over here that does not contain the regulatory CD3 promoter. Okay, so let's go to question B. Okay, question B. The response indicate, remember that we're talking about independent variable, so the response indicates that the independent variable is the particular plasmid or that it is the CD3 gamma regulatory sequence introduced into the lymphocytes. So the independent variables are each one of these fragments. Okay, they're independent. And what would the dependent variable be? The relative amount of luciferase that's being expressed in that T lymphocyte. So these are all independent, but they affect the dependent variable, which is relative luciferase activity. Okay? Identify the plasmid that was used as a negative control for luciferase activity. So let's look at that. Question B2. The response indicates that the plasmid used as a negative control was a parent plasmid, or that it was a plasmid without any regulatory sequences inserted. So all of these couldn't have been our negative control because all of these have a sequence. They have the CD3 regulatory sequence. So what could have been a negative control? Either the parent plasmid, which is a plasmid that has not been inserted any of these sequences, okay? Or a parent plasmid with non-CD3 active promoter. So it doesn't have any of the fragments and it also doesn't have the promoter. Okay, this one's a little bit unclear for me because I am thinking that these are very, very similar. But what I'm thinking is that this parent plasmid, actually, that's what I'm telling you. These two are very similar to me. I would think that this parent plasmid over here would also have to have the absence of the CD3 active promoter. And I also think that that's why either one of these two are fine as a negative control. So if you wrote either one of these two parent plasmids, you guys got this correct. Okay? Okay. And finally, question B, the last part of question B, that says, the response indicates that the plasmid used, question B3. Okay. The response indicates that the plasmid with non-CD3 gamma active promoter is a positive control for the effective introduction of the plasmids into the T lymphocytes and for the successful use of the luciferase assay to measure promoter activity. So let me go right back to think about how that relates to question B. Justify including the plasmid with a non-CD3 active promoter in the experiments. Right, you will include this non-CD3 active promoter in the experiment because you kind of want to know, wanted to know how all of the other sequences affected the amount of luciferase activity. But when you're including a plasmid that does not have a regulatory genetic chain, which is the CD3, what you're going to expect is that the luciferase activity is going to just be extremely high because in these sequences, since they contain negative regulatory sequences, then they're going to inhibit, at least in some extent, the luciferase activity. But in this case, since you're not including any of the sequences, you're allowing this enzyme to simply transcribe and be expressed to its full potential. So a lot of it. So that's also why it's a positive control, because in this case, you're checking out what's happening when you have absolutely no regulation. Okay. So hopefully that makes sense. That was a very difficult question, but hopefully it made sense. Okay, so part C. Sorry, let's go to part C. Identify the plasmid that must contain the CD3 core promoter sequence, but the fewest or no negative regulatory sequences. Based on the data in Figure 2, describe the most likely cause of the variation in luciferase activity among the cells containing plasmids 419, 309, 239, and 199. Calculate the approximate percent increase in luciferase activity between cells containing plasmid 59 and 149. Round to the nearest whole number. Okay, so let's start with identify the plasmid that must contain CD3 core promoter sequence, but the fewest or no negative regulatory sequences. We're going to go into question C1. The response indicates that the plasmid that must contain core promoter sequences, but the fewest or no negative regulatory sequences. No negative, remember negative. So there's very few things inhibiting the transcription and expression of the bioluminescence gene. Has to be the 15. So let's go back to 15 and see why. Right over here, 15. Okay, so all of these are the same except for having different fragments, right? Okay, now the one that contains the fragment 15 is the one that's producing the most luciferase. What does that tell you? That tells you that this one is going to be the regulatory sequence that contains the least inhibitory or negative regulatory sequences. They can contain positive and negative. So for example, this one over here probably contains the most, or this one, sorry, this one over here, the 59, probably is the one that contains the most negative regulatory sequences, because look at this, it results in the production of very little luciferase, rather than this one that results in the most production of luciferase, of course not taking into account the positive control, because this one does not contain the actual regulatory sequence. It contains no sequences. Okay, hopefully that makes sense. Okay, then in C2, the response indicates that the different sequences cloned into the plasmid contain different combinations of positive and negative regulatory sequences. So if you do not remember what the question was, it was simply describe the most likely cause of variation in all of these different sequences. And the only differences in these sequences is how many positive or how many negative regulatory sequences it contains. And that's going to influence how much luciferase is produced. Okay? Then it says calculate the approximate percent increase in luciferase activity between cells containing the 59 and the 149. So let's see. 59 right over here and 149 right over here. 59 goes around 2, 149 goes around 4. So 2 to get to 4, it is an 100% increase, right? It increases by 2. So it's 100% increase. And if we check that out in question C, the response indicates that the percent increase is approximately 100%. The 59 ends at 2, a luciferase activity. The 149 ends at 4, luciferase activity. And simply because to get from 2 to 4, there's an addition of an extra 2, that is 100% increase. Okay, and let's finish with question D. Sorry. Okay, predict the most likely observed level of luciferase activity if plasmid 789 is introduced into non-lymphoid cells. So non-lymphoid cells, not the T lymphocytes. And where do you introduce it? Let's say a line of kidney tissue cells. Provide reasoning to justify your prediction. So every time that they're telling you to predict something, let's go back to the text. Okay? And you're going to see that at the very beginning it says CD3, which is this regulatory sequence, is a signaling protein, or CD3 is a signaling protein that goes into the sequence, that's expressed by the sequence, is a signaling protein that is typically found only in the plasma membrane of immune system T lymphocytes. Okay, so what does that mean? If you insert this gene sequence into a cell that is not an immune system cell, you will not be able to produce the signaling protein CD3. You cannot produce it because it is not found. Your raw material will be not found in other cells. Okay, so this one was an actual pretty easy answer if you think about it. What would the answer simply be? The response indicate that the plasmid that must contain the CD3 core promoter sequence. Oh, sorry, no, I'm completely incorrect. This is question D. I'm so sorry if I got you confused. Okay, the response indicates that there will be little or no luciferase activity in the 789 if 789 is introduced in non-lymphoid cells such as lines of kidney tissue cells, because they do not have the ability to produce the signaling protein. Okay, and the last bit, question D. There. Provide reasoning to justify your prediction. Okay. The response indicates that the non-lymphoid cells will not contain the transcription factors that bind to the CD3 gene regulatory sequences. So if the cell is not able to produce the protein, it is because it doesn't have the mechanisms to start transcription of that gene, okay? So hopefully that made sense. I know this is a super long video. I apologize. It is an extremely difficult question, but we need to do the work. We need to put in the work of being able to get to this level, okay? So I'm going to allow you guys now to go into Schoology, to go into the assignment of the scoring assignment. You're going to have a link to this document, and you're going to score yourself, and you're going to tell me where you got your mistakes. You're also going to paste your original answers into each one of the questions, like in section A, section B, section C, so that I can go through your reasoning as to why you're grading yourself in that way. I hope this is helpful. Please message me if you have any questions. Thank you.